The market for gene therapies continues to grow, and the need for high-quality plasmid DNA is increasing. My name is Nate Paulin, and I'm delighted to welcome you to this product demonstration on the PATfix pDNA analytical platform. Many companies are looking to insource pDNA production and purification, usually as a key starting raw material for subsequent viral vector and mRNA production and purification. Our extensive experience of working with numerous companies involved in the production of plasmid DNA for over 20 years has enabled us to gain valuable insights into the key challenges our clients face in their plasmid DNA production processes. One of the most common issues we face is the inability to perform analysis on lysate samples in the pre-capture step of plasmid DNA production due to the complexity of the lysate samples. Many are familiar with lab-scale plasmid isolation via spin column kits. However, knowledge gained from lab kits is difficult to implement in a large-scale pDNA purification process. As a result, our clients have been unable to obtain crucial data regarding the plasmid DNA content within the bacterial cells. Furthermore, the absence of this information has hindered efforts to optimize the lysis process, which is recognized as a pivotal step in the recovery of plasmid DNA from bacterial cells and in maintaining its purity. Another significant challenge we have identified through our work with a number of customers is the lack of effective quantification techniques for determining pDNA concentration. The most commonly used method for determining pDNA concentration is nanodrop. While it is a rapid spectroscopic method that requires a minimal amount of sample, it can also lead to inaccurate measurements and errors. Firstly, nanodrop is unable to distinguish between the different nucleic acids commonly present in pDNA-related samples, such as host cell RNA and DNA. It can also give false quantification results due to the presence of many impurities that can absorb UV light. This can lead to errors in the recovery of the purification process. Second drawback of this type of spectroscopic method for nucleic acid quantification is the influence of pH and conductivity of the matrix in which the pDNA concentration is determined. This phenomenon can also contribute to inaccurate determination of pDNA concentration. A further limitation of this technique is its inability to distinguish between different pDNA isoforms. The objective at the end of the purification process is typically to obtain a single pDNA isoform. The proportion of individual pDNA isoform may vary throughout the purification process due to the inherent instability of pDNA. A lack of knowledge regarding the isoform composition of pDNA and the presence of contaminants, such as genomic DNA, may result in complication when utilizing the pDNA in subsequent processes. An existing solution to this issue is the utilization of electrophoretic techniques such as agarose gel electrophoresis and capillary gel electrophoresis. However, the primary limitation of agarose gel electrophoresis is its inability to quantify pDNA isoforms, while the use of capillary electrophoresis is challenging and requires a great deal of experience. The shortcomings of conventional pDNA analytical techniques prompt us to develop a streamlined, single-method chromatographic solution for pDNA characterization. The main objectives we sought to achieve during the development of a method for pDNA characterization were the ability to robustly analyze all samples involved in the pDNA process, the capability to distinguish between all possible pDNA isoforms, and the capacity to quantify and monitor all contaminants present in different pDNA samples in addition to individual pDNA isoform quantification. Furthermore, we aim to ensure that the analytical method is straightforward in terms of sample preparation, processing, and interpreting the results. We are pleased to present our solution, the chromatographic PATfix pDNA analytical platform, which has been specifically designed for the analysis of pDNA samples at all stages of the pDNA process. The process of developing the method commenced with the selection of an appropriate chromatographic medium. We identify a solution in the use of a weak anion exchanger CMAC pDNA column with 1.4 micrometer channels, which we produce in-house. This column enables the separation of all essential pDNA isoforms, the differentiation of pDNA from other impurities in the sample, and the optimal recovery of pDNA from the column with no carryover. It is compatible with up to 4 molar salt buffers and 1 molar sodium hydroxide for sanitizing the column. Once the appropriate chromatographic medium had been identified, we proceeded with the optimization of buffer composition and gradients. Our objective was to optimize the method in order to achieve the best possible resolution between pDNA isoforms and other contaminants, to provide the best possible recovery and to minimize sample carryover. The objective was to ensure that the method could be applied to all types of samples encountered in the pDNA production process, from host cell lysates to purified pDNA samples. The full potential of the PATfix pDNA method is revealed in lysis host cell samples. The process effectively separates all contaminants, proteins, host cell debris, host cell RNA, and host cell DNA from target pDNA isoform. The separation of pDNA is also achieved, enabling the quantity of pDNA to be determined throughout the entire production. Moreover, this method enables the tracking of contaminants throughout the entire purification process. This analytical method is also able to distinguish between different pDNA isoforms. In this chromatogram, the separation of the 3 pDNA isoforms is demonstrated. The 3 isoforms are clearly distinguishable from one another. This feature of the method is particularly useful for monitoring and optimizing the linearization reaction. Once developed, the pDNA analytical platform was tested on a wide variety of pDNA samples, including small and large plasmids, plasmids for IVT reaction, and nanoplasmids. However, we encounter issues with the poor plasmid recovery when analyzing larger plasmids with the CMAC pDNA column with 1.4 micrometer channels. To address this issue, we have developed a new CMAC pDNA analytical column with larger 6 micrometer channels. Larger channel size has enabled us to resolve the poor recovery issue for larger pDNA isoforms. The two methods for small and big plasmids were validated on all important parameters, including LOD and LOQ, linearity, accuracy, precision, and robustness. All validated parameters comply with the criteria set on the ICH for biological analytical method validation. During the development of the pDNA analytical platform, we encountered another significant challenge. In discussions with a number of clients, the majority have highlighted difficulties in setting up a chromatographic analysis at their site, citing a lack of knowledge and chromatographically educated personnel as a key issue. To address this challenge, we have streamlined the process to make the pDNA chromatographic analysis more straightforward and intuitive for our clients. The first step was to develop new PATfix software that is much more straightforward than conventional HPLC software and easy to use. The software also allows complete control over the PATfix hardware and detectors. We have implemented a comprehensive step-by-step guide for the pDNA platform, ensuring that even those without prior experience can perform analytics with ease. In conclusion, the PATfix pDNA analytical platform provides a comprehensive and robust solution for the accurate characterization and quantification of plasmid DNA across all stages of production. By overcoming the limitation of traditional method, it ensures precise separation of pDNA isoforms and contaminants, making it an essential tool for optimizing pDNA processes. The PATfix platform is accessible to all, even those with limited chromatographic experience, thanks to its user-friendly software and streamlined protocols. This empowers companies to efficiently insource their pDNA production and purification. Thank you for participating in this demonstration. We hope that you gain a better understanding of the PATfix pDNA analytical platform and how it can benefit your manufacturing processes. If you are interested in learning more about the PATfix pDNA analytical platform, please visit our website or contact our sales team for a personal consultation. We also invite you to download and try our PATfix demo software, available on our website. Let's work together to take your pDNA analysis and production to next level.